Hello everyone. Today we will study cell cycle and cell division in detail. Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. Cell division, cell growth and DNA replication have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesis the other constituents of the cell, and eventually divides into two daughter cells is termed cell cycle. Cell growth is a continuous process, but DNA synthesis occurs only during the synthesis phase S phase of the cell cycle. During cell division, replicated chromosomes DNA are distributed to daughter, daughter nuclei by a complex series of events. These events are under the genetic control. Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in every 24 hours. Time period of cell cycle varies from organism to organism and also from cell type to cell type. Yeast can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two main phases: interphase, M phase, also called mitosis phase. Interphase. It is the phase between two successive M phases. The interphase is called the resting phase. The interphase lasts more than 95% of the duration of cell cycle. In this phase, the cell prepa- prepares for division by undergoing both cell growth and DNA replication in orderly manner. The interphase is divided into three phases: G1, gap 1 phase. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During this phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S phase, also called synthesis phase. S phase marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. But there is no increase in the number of chromosomes. If the cell had diploid or 2n numbers of chromosomes at G1, then even after S phase, the number of chromosomes remains the same, that is 2n. In animal cells during the S phase, DNA replication begins in the nucleus and centriole duplicates in the cytoplasm. G2, gap 2 phase. Final preparation of M phase occurs during this phase. In G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Interphase is the longest and very active phase of cell cycle. Point 2. During G1 phase, cells synthesize different types of RNA and proteins. Point 3. Quiescent stage, also called G0 stage. Some cells in the adult animals do not appear to exhibit division, example heart cells, and many others divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells do not divide further. They exit G1 phase to enter an inactive stage called quiescent stage of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. M phase, also called mitosis phase. M phase represents the phase when actual cell division or mitosis occurs. In the 24-hour average duration of cell cycle of a human cell, cell division proper lasts for only about an hour. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes karyokinesis and usually ends with the division of cytoplasm called cytokinesis. In animals, mitotic cell division is only seen in the diploid somatic cells, exception male honey bees that are haploid. Plants show mitotic division in both haploid and diploid cells. M phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. The numbers of chromosomes in parent and progeny cell remain the same, so it is also called equational division. Cell division is a progressive process and very clear-cut lines cannot drawn between various stages. Although for convenience, mitosis is divided into following four stages of nuclear division called karyokinesis. Number 1, prophase. Number 2, metaphase. Number 3, anaphase. Number 4, telophase. Prophase. Prophase is the first stage of mitosis which follows the S and G2 phase of interphase. The new DNA molecules formed during the S phase are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which is duplicated during S phase of interphase, now begins to move towards the opposite poles of the cell. The completion of prophase is marked by the following characteristic events. A. Chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at the centromere. B. Centrosome begins to move towards opposite poles. Each centrosome radiates out microtubules called esters. The two esters together with spindle fibers form mitotic apparatus. C. Cell at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and the nuclear membrane. Metaphase. The complete disintegration of the nuclear envelope marks the start of the metaphase. The chromosomes are spread throughout the cytoplasm of the cell. By this stage, condensation of chromosome is completed, and they can be seen clearly under the microscope. This is the stage at which morphology of chromosomes is most easily studied. Metaphase chromosome is made up of two sister chromatids, which are held together by the centromere. A small disc-shaped structure at the surface of the centromeres are called kinetochores. These structures serve as the sites of the attachment of spindle fibers. Metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole and its sister chromatids connected by its kinetochore to spindle fibers from opposite poles. The plane of alignment of the chromosomes at metaphase is referred to as metaphase plate. The key features of metaphase are A. Spindle fibers attached to kinetochores of chromosomes. B. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both poles. Point 3. Anaphase. Each chromosome arranged at the metaphase plate is split simultaneously and the two daughter chromatids, now referred to as chromosomes of the future daughter nuclei, begin their migration towards the two opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome is directed towards the pole and hence at the leading edge with the arms of the chromosome trailing behind. The anaphase is characterized by following key events. A. Centromeres split and chromatids separate. B. Chromatids, now called chromosomes, move to opposite poles. Different shapes of chromosomes may be observed at anaphase V or J or L or I-shaped, depending upon position of kinetochore or centromere. Types of chromosome: metacentric V-shaped, centromere position median. Submetacentric L-shaped, centromere position submedian. Acrocentric J-shaped, centromere position subterminal. Telocentric I-shaped, centromere position terminal. Fourth, telophase. At the beginning of telophase of the chromosomes that have reached their respective poles decondense and lost their individuality. The individual chromosomes can no longer be seen and each set of chromatin material tends to collect at a mass in the two poles. The telophase is characterized by following key events. A. Chromosomes cluster at opposite spindle poles and their identity is lost as discrete elements. B. Nuclear envelope develops around the chromosome cluster at each pole forming two daughter nuclei. C. Nucleolus, Golgi complex and ER reform. Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by a separate process called cytokinesis, at the end of which cell division is completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation starts in the center of the cell and grows outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate that retains the middle lamella between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate conditions arises, leading to the formation of syncytium, example liquid endosperm of coconut. Cytokinesis is the last step of M phase. It starts towards the middle of the anaphase and is completed with the telophase. A mitosis is a simple method of direct division characterized by splitting of nucleus followed by that of cytoplasm. In this method, the nucleus elongates and constricts in the middle to form two daughter nuclei. This is followed by a centripetal constriction of the cytoplasm to form two daughter cells. It occurs in meganucleus of paramecium, nuclei of internodal cells of Cara, aging and deceased cells, etc. All those substances or chemicals which prevent the cells from entering into the normal mitotic divisions are called mitotic poisons. The enzyme ribonuclease acts as poison at prophase. Azide and cyanide also inhibit prophase. Mustard gas causes fragmentation and agglutination of chromosomes. The alkaloid colchicine inhibits the formation of mitotic spindle, inhibits polymerization of microtubules and holds the cells in metaphase. Significance of mitosis. Mitosis is of great significance in the life of the organism. Number 1. Growth. Somatic cells are formed by mitosis. Therefore, it is essential for growth and development of multicellular organisms. Number 2. Maintenance of surface volume ratio. An overgrown somatic cells is induced to divide so that mitosis helps in maintaining a proper surface volume ratio. Number 3. Maintenance of chromosome number. Mitosis involves equitable distribution of all chromosomes so that all the cells of multicellular organisms have the same number and type of chromosomes. Number 4. Repair and regeneration. It is a mechanism of replacing old or worn out cells and also helps in regenerating part or whole of the organism. Number 5. Reproduction. It is a method of multiplication of unicellular organisms. Number 6. Healing. An injury is healed by repeated mitotic division of the surrounding cells.